Welcome back. So, have you ever stopped to think about how our balance system can pick up on the tiniest little head tilt, but also handle a sudden, rapid spin without just completely shorting out? Well, the secret is buried deep inside the tiny pores of our inner ear hair cells. In this explainer, we're going to crack the case on the proteins that actually build these pores, TMC1 and TMC2, and see how their partnership is the key to our vestibular system's incredible range. You know, for decades, this channel was basically a black box. We knew it existed, we knew what it did, but what it was actually made of, that was a huge mystery in neuroscience. So how did we finally figure it out? Well, it was like a great molecular detective story. Let's follow the clues. And look, this isn't just some academic puzzle. It's a question that's fundamental to what we see in the clinic. Just picture a vestibular hair cell. It's got to be sensitive enough to notice a whisper of movement, but also tough enough to not get overwhelmed by a big, jarring motion. How on earth does it pull off that kind of dynamic range? The answer, it turns out, is literally built into the pore itself. So let's get into the investigation. How did researchers finally unmask the proteins running the show? It wasn't one single aha moment. It was more like 3 different lines of evidence all converging, all pointing to the exact same suspects. The case was really built on these 3 pillars. First, they used genetics, just completely remove the suspect genes. Second, they got clever and looked at a very specific mutation. And third, they used structural data to see how all the pieces physically fit together. All right, first piece of evidence, the genetic knockout. And what's so striking here is just how clear the result is. In a normal hair cell, you stimulate it, you get an electrical current. Simple. But if you take out the genes for TMC1 and TMC2, poof, the current is just gone. The takeaway is about as blunt as it gets. No TMC proteins, no mechanosensitivity. It proves they are absolutely essential. Now, the second piece of evidence is maybe even more powerful. It's a real smoking gun. It all comes from this one single-point mutation in the TMC1 gene. It's famously called the Beethoven mutation because of its connection to deafness. What researchers found was that this one tiny little change to the protein completely altered the fundamental properties of the pore, like how many ions could flow through and even which ions it preferred. And this right here is the critical logic. If making a tiny change to the protein directly changes how the pore works, it's incredibly strong evidence that the protein isn't just helping out on the sidelines. It means the protein is a core part of the pore itself. And the final nail in the coffin, so to speak, came from structural biology. These studies basically gave us the blueprints, showing a model where the TMC proteins pair up. They form these dimers to actually build the channel. This physical data just fit perfectly with all the functional data from the knockout and mutation studies. All the pieces clicked. So with that, case closed. All the evidence pointed straight at 2 culprits, transmembrane channel-like proteins 1 and 2, or as we know them, TMC1 and TMC2. These are the essential guys, the pore-forming subunits we've been looking for. Okay, but solving one mystery just opens up another one, right? Why 2 different subunits? Why have both TMC1 and TMC2? Well, it turns out that having this tag team is the secret sauce behind the vestibular system's incredible performance. Let's dig into why. This is where we see a really important split between our hearing and balance systems. In the cochlea, the hair cells basically grow out of needing TMC2. Its expression fades after early development, leaving the adult cells mostly with TMC1. But the vestibular system? Totally different story. It holds on to both TMC1 and TMC2 all the way into adulthood, and that's no accident. It's absolutely central to its job. So why keep both around? Well, because they're specialists. They have different strengths. Think of a channel with a TMC2 subunit as the high-sensitivity, high-flow specialist. It lets more ions through. A TMC1 channel, on the other hand, is more of your stable, reliable workhorse, lower flow, but rock solid. Now let's connect this right back to the problem we started with. The vestibular system has to deal with this ridiculously wide range of inputs, everything from the tiniest, most subtle head movements to really intense high-frequency accelerations. And here it is. This is the big insight. By having both TMC1 and TMC2 in its toolkit, a vestibular hair cell can mix and match, building a whole population of channels with slightly different properties. It's this molecular variety that gives the entire system its amazing dynamic range, making it both incredibly sensitive and incredibly resilient. Yeah, molecular tuning is the perfect way to think about it.
The cell is blending these subunits to get the best of both worlds. You've got TMC2 providing that exquisite sensitivity for the really subtle stuff, while the more stable TMC1 channels act as the backbone, making sure the system doesn't just get blown out by large accelerations.
Okay, so let's wrap this up. What are the big picture clinical takeaways you really need to remember from all this?
All right, 3 things. First, the big one. TMC1 and TMC2 are, without a doubt, the proteins that form the pore of the MET channel. Mystery solved. Second, a channel's identity predicts its function. If you know whether it's built with TMC1 or TMC2, you have a really good idea of how it's going to behave. And third, and this is crucial for us, this co-expression of both proteins is the key reason our vestibular system has such a broad operating range, which is absolutely essential for everyday function.
So now that we've identified the pore, a new question pops up. We are always under the force of gravity. It's a constant stimulus. So why don't these channels just get jammed open all the time, making us unable to sense any new movements? So we've figured out how TMC1 and TMC2 build the MET pore and give our balance system its incredible range. But in our next explainer, video 7, we're going to tackle that saturation problem. We'll explore the really clever adaptation mechanisms that keep our hair cells from being overwhelmed by gravity, and we'll see how this connects directly to the different kinds of vestibular complaints we see in patients. You won't want to miss it. Thank you so much for joining me for this part of the series. To explore more topics on dizziness, vertigo, and imbalance, or to get clinician and patient resources, please visit us at australiandizzinessclinics.com. And of course, don't forget to subscribe and leave a rating.